Okay. So, good day, class. This would be the first lesson for the finals. We have your lesson 8, Respiratory and Childhood Viruses. So, when we talk about respiratory disease class, please take note that this would account for an estimated 75 to 80% of all acute morbidity. When you say acute morbidity class, when you say morbidity, this refers to individuals with the disease. So 75 to 80% of acute individuals or acute or people with acute diseases are in the form of respiratory disease. And most of these illnesses are in the form of viral infection, meaning class that most of these acute illnesses would come in the form of viral respiratory infections. That's why it's very easy. It's very easy to acquire viral diseases, especially viral respiratory infections. They are the ones that are most common. Now, although a majority of the episodes may not require medical attention, the overall average is 3 to 4 illnesses per year per person. Although the incidence varies inversely with age, the morbidity is significantly higher in elderly population. So, as you've learned in your immunology and serology, elderly people would tend to have a weaker they tend to have a weaker immune system. Thus, it's easier for them to acquire this type of illnesses. Then another factor to consider is seasonality. So incidence is lowest in summer months. So when you say incidence, class, just to give you a recap, incidence refers to new cases. So new cases of respiratory viral diseases are lowest in summer months. Remember, in summer months, class, the temperature are hotter, while incidence or new cases are higher in winter. So as a defining characteristic of viral respiratory diseases, they are plenty or they tend to thrive more or they tend to survive more in cold temperature. Because remember, class, diba, if what you learned in your introduction to virology, viruses, class, are heat labile. They are susceptible to heat. They are easily destroyed by heat. That's why viral respiratory diseases are more common in colder temperature or in seasons such as winter. And in the Philippines, we don't have a winter season, but we do have a rainy season. So in rainy season, the viral infections are more common, especially viral respiratory diseases. In addition to the ability to cause a variety of acute respiratory disease syndrome, this group of viruses would relatively share a short incubation period of 1 to 4 days and has a person to person mode of spread. So the reason why, class, there's a very high amount of cases for respiratory viral disease is because of this. They have a relatively short incubation period of 1 to 4 days, meaning, class, the symptoms would manifest already within 1 to 4 days, and they have a very easy mode of transmission, which is person to person mode of spreading. So imagine if you're commuting via the train station or via train or commuting via public transport, and you have a person that's infected with a viral respiratory disease. If they do not practice proper hygiene, let's say they sneeze and they tried covering it using their hands, you could easily and when they touch the public transport, you could easily acquire the virus. So that's one way. That's one of the reason why it has a high percentage of morbidity or active cases. Now they are transmitted via direct transmission, via infected droplet nuclei or indirect, and by hand transfer of contaminated secretions to nasal or conjunctival epithelium. So these viruses, class, these respiratory viral diseases are transmitted via secreted secretions, nasal secretions. So to give you an example, when a person sneezes or coughs, usually these are via nasal droplets. Now, these respiratory viral agents are associated with an increased risk of bacterial superinfection of damaged tissue of the respiratory tract, and all have a worldwide distribution. So when you acquire this type of viral diseases class, you're more prone to bacterial superinfection. An example of a bacterial superinfection is your pneumonia. Imagine having a basic type of respiratory disease, a mild flu or a mild cough. If you do not treat it immediately, you allowed it to become severe, you could easily be affected by other bacteria that could cause pneumonia. And this could lead to a very difficult case of pneumonia, respiratory infection disease. And another thing to consider is that they have a worldwide distribution, so it's very common in the entire world. Let's start with the first group of virus class, respiratory virus. Let's start with your Parvoviridae. So here on the left side, you'll see a table summarizing the details of this virus. So kindly memorize them, class. They will come out in your quiz and your exam. So let's start with your Parvoviridae. So this is a family of viruses known as your Parvoviridae. The common name is the Parvovirus. And the topic that we would target here is your Parvovirus B19. Now, as if you're a person that loves dogs, you would often hear parvoviruses. Diba? If you're familiar, if your dog becomes weak, it has a bloody diarrhea and would often vomit yellowish vomit class. It means that your dog may have parvovirus infection. Now we're going to tackle here the only known parvovirus affecting humans, parvovirus B19. Now, the characteristic of this virus is that this is a single-stranded DNA virus. It has a icosahedral capsid. It has no envelope, or it's a non-envelope virus. And the one that we're going to tackle, as I mentioned, is parvovirus V19, which is the only known human pathogen. So you could acquire this via close contact and via respiratory transmission. The disease known it causes is erythema infectiosum, or also known as the fifth disease. So erythema infectiosum is also known as your fifth disease. It's commonly seen or seen in aplastic crisis in patients with chronic hemolytic anemia. So when you say aplastic crisis, this is the one that would usually affect your bone marrow class. What happens in a plastic crisis is that your bone marrow will no longer produce red blood cells. And remember, the function of your red blood cells is to carry or transport oxygen to the different tissues and organs of the body. So if there's no red blood cell, there would be hypoxia, or there would be decreased oxygen level. So these are seen in patients with chronic hemolytic anemia, fetal infection, and stillbirth. To detect this type of virus, you would use serology, PCR, and even biopsy. Treatment is supportive. Prevention is avoiding contact. So that's the summary class of your parvoviridae. Now let's discuss them in a deeper discussion. So we have your parvovirus. They came from the Latin word parvus, parvus meaning small. It is a widely distributed among warm-blooded animals. So we have here our target virus, parvovirus B19. This represents the one human pathogen in the family. So it's the only parvoviridae virus known to affect humans. Its replication in human cells is restricted to the erythroid progenitor cells. So if you're familiar with your hematopoiesis, diba class? In your hematopoiesis, you all have what we call stem cells. Now, these stem cells class would give rise to different progenitor cells. We have your erythroid progenitor cell, your myeloid progenitor cell, something like that. So, the target here is the one that it would usually affect is the erythroid progenitor cells, making the adult bone marrow and fetal liver, which is the site of erythropoiesis during fetal development, the major sites of viral replication. If you still remember in your hematology 1, aside from the bone marrow, during fetal development, the liver is also one of the sites of RBC production. But as a fetus matures, nagiging teenager, it becomes he or she becomes a teenager, an adult, it starts becoming the bone marrow only that would produce the erythroid cells. So the ones affected here are the fetal liver and the fetal bone marrow, which becomes the major sites of viral replication. The virus is spread through the respiratory route and is characterized by fever and a unique slap cheek rash. So if you take a look at the picture on the lower right side, class, you would see a fetus or a child affected with fifth disease or your erythema infectiosum. It has a slap cheek appearance. Now, important disease caused by B19 would include the fifth disease, or this is one of the fifth of the childhood insantemas, a plastic crisis in patients with underlying hemoglobinopathies, and fetal infection, hydrops fetalis, resulting from transplacental transmission. Now, parvovirus would cause a basic illness in humans. So it causes two phases of diseases in humans. So the first phase class would comprise of fever, malaise, myalgia, and chills, which corresponds to peak levels of virus destruction or erythroblast. So in the first phase class, when your virus is being destroyed by your immune system, an effect of this viral destruction would be fever, malaise, myalgia, and chills. This phase, when mild, may be overlooked class. So when the phase is mild, it can be overlooked or considered non-specific viral disease. Now, after the first phase, it now develops into the second phase, wherein a rash and arthralgia would occur, having after the virus has disappeared at a time when the parvovirus specific antibody can be detected. This is consistent with the conclusion that the rash is caused by immune complexes in the capillary of the skin. So, for you to understand, let's go back to the previous slide. One characteristic class of having erythema infectiosum is this so-called slap cheek appearance. Now, in the second phase class, there would be arthralgia and there would be the removal, the removal of the virus. If you recall, in the first phase, the virus are being destroyed. Once it reaches the second phase, the virus is already removed. Now, what you would use to detect the infection or the disease is by identifying the antibodies. Now, the reason, as mentioned in the statement earlier, is that in the second phase, antibodies would be detected in the capillaries. That's why if these antibodies would develop or would deposit in the capillaries, it would now form this red cheek appearance of the child. So ito siya, class. So the rash is caused by an immune complex in the capillaries of the skin. Now, how would you diagnose this parvovirus B19? So specific IgM or virus-specific IgG antibody testing with acute and convalescent sera is used. Parvovirus cannot be cultivated in usual cells available in clinical virology lab. So what you need to do to test this is via serology, class, specifically via identifying IgM or IgG antibodies. Okay, let's continue. Now let's go to the second virus here, Adenoviridae. So you also have here the summary of your Adenoviridae. So the family here is your Adenoviridae. The common name is the adenovirus. So here our target focus would be the adenovirus. So the characteristic of your adenovirus is that this is a double-stranded DNA virus. It has a icosahedral capsid. It has no envelope. Approximately there are 50 human serotypes. So remember, class, there are 50 types of adenovirus. You have your adenovirus 1, 2, 3 until 50. Transmission is via respiratory. It could also be via fecal oral route and even direct contact through the eye. Now, disease, it's known to cause respiratory and gastrointestinal disease. So, the respiratory diseases would include your pharyngitis, pharyngoconjunctival fever, keratoconjunctivitis, pneumonia, hemorrhagic cystitis, disseminated disease, and it would cause gastrointestinal disease in the form of gastroenteritis in children. Detection of this virus class is done via cell culture using Hep2 and others continuous human epithelial lines. Enzyme immunoassay could also be used for gastroenteritis serotypes 40 to 41. Treatment is supportive. Prevention, there are vaccines for serotypes 4 and 7. So that's the summary of your adenoviridae. Let's have a deeper discussion. So adenovirus class is the member of the family Adenoviridae and the genus Mastodenovirus. This should be E. So the genus is your Mastodenovirus. The virus has a linear double-stranded DNA, icosahedral symmetry, and a size of 70 to 90 nanometer. Adenoviruses were first isolated from human adenoid tissues. At present, approximately 50 serotypes of human adenoviruses have been described. However, most disease is associated with only third of these types. Adenoviruses would cause less than 5% of all acute respiratory disease in the general population. In addition, class adenoviruses serotypes 40 and 41 would cause gastroenteritis in infants and young children. Other disease would occur but is less common. Specimens to be collected would include throat swab, nasal washing, conjunctival swab, scrapings, or feces. So take note, class, of the percentage of respiratory diseases that it would cause or the population would affect. It's only less than 5% in the general population. So it's a very low percentage class. Then take note also that serotypes 40 and 41 are known to cause gastroenteritis in infants and young children. Also take note of the tissues affected, specifically the human adenoid tissue. So the human adenoid tissue is located at the back of your throat, just above your tonsils, class. So if this is your tonsils, it's located above here, just at the side of your ear. That's why it can affect the eye also. Now your diagnosis is via conventional cell culture using Hep2 cells and serologic methods. Let's go to the third type of virus. You have your Poxviridae. So here we also have another summary class. So the family is Poxviridae. The common name is poxvirus. So the viruses included here are your smallpox, your molluscum contagiosum, and your orthovirus. So the characteristic of your poxvirus class is that this is the largest and most complex of all viruses. So this family class is the largest in size and most complex of all viruses. This is a brick-shaped virion with no conforming symmetry. So when you say no conforming symmetry, if you remember, it is considered complex. And then when it's complex class, it has a variable shape, meaning the shape would change. It has a double-stranded DNA genome. Transmission is by respiratory droplets for smallpox, while direct contact would be molluscum and orf. Diseases class. It's known to cause all diseases of the skin, specifically smallpox, generalized infection with a pustular rash. If you remember in your immunoserology, during the early times, they would try to induce immunity. They did an experiment on how to induce immunity class using smallpox through the method known as variolation. So what they would do, they would get the pus, yung pustule. So imagine this is your arm, there would be rashes here. So what the doctors or what the experts during those times did is that they would poke the rash, and inside the rash there would be pus. Pus in Tagalog is nana. So may nana or may pus yan. After poking the pus, they would get it and they would introduce it. They would introduce it through the nose. And it was believed that it would induce immunity. Now, please take note, class, that smallpox and chickenpox are different, as the former is extinct. So the former here is your smallpox. Smallpox is already extinct, class. It was extinct because of the contribution of Edward Jenner. If you remember Edward Jenner in your Immunocero, he was the one that would introduce vaccination. So because of vaccination, smallpox was eradicated or extinct. Now, molluscum would manifest as benign modules of the skin, and ORP would manifest as localized papules or vesicle of the skin. So detection is via EM, electron microscopy of material from skin lesions. Treatment is supportive. Prevention is vaccine for smallpox, and avoid contact with infected individuals. So let's have a deeper discussion. So the pox viruses are the largest and most complex of all viruses. It has a size of 225 by 300 nanometers. These are DNA containing viruses that are enveloped with complex coats, with brick shape and complex morphology. The family would include variola. Under variola, we have the smallpox virus, vaccinia virus, as well as the agents of cowpox, monkeypox, and canary pox. So cowpox would usually affect bovine animals, monkeypox would affect monkeys, and canary would usually affect birds. So these are the zoonotic forms of your smallpox: cowpox, monkeypox, and canary pox. So diseases caused by pox virus would include smallpox. This is a devastating and frequently fatal disease of historical importance. It's very important in history class because it was used to develop your vaccination. Eliminated from the world in 1980. Then we have your molluscum contagiosum. This is an exclusively human disease of the skin that is self-limited but may recur during periods of severe immunosuppression. And we have ORF. ORF is a localized infection of the skin caused by viruses responsible for dermatitis in sheep, goats, and related animals. Okay, let's continue. So this is the appearance class of your pox viruses. So take note of the varying shape and sizes. So as I mentioned last time, it has a complex. It has a complex shape. It has a variable shape. So it would be in a brick shape. It could be in different shapes. So here are some of the genuses of your Poxviridae. We have your genus Orthopoxvirus and the genus Parapoxvirus. And the thing you need to consider about this virus is that it has an envelope. Compared with your adenovirus, your parvovirus, which are non-envelope. This one, your Poxviridae, is envelope. Let's go to the fourth one, very important virus class of the respiratory diseases. We have your Orthomyxoviridae. So there's also a summary here. Now, the thing you need to consider about Orthomyxoviridae is that they're known to cause your influenza. So the family is Orthomyxoviridae. The common name is the Orthomyxovirus. So the Orthomyxovirus is our target now. So the characteristic of is that this is segmented. It has 8 separate molecules. It is a single-stranded RNA genome, helical capsid with envelope, 3 major antigenic types: influenza A, B, and Z. Types A and B would cause nearly all human disease or all human influenza. Basically, class influenza C is not relevant. A and B are the ones that are deadly and can cause disease in humans. Transmission is contact with respiratory secretion. So let's look at the summary of each influenza. So for influenza A, it's known to cause your malaise, headache, myalgia, and cough. Primary influenza pneumonia in children, bronchiolitis, croup, and otitis media. So in children class, it would manifest bronchiolitis, croup, and otitis media. But in primary pneumonia, Malay headache, the problem with this type of disease class is that there's really no treatment. You need to do supportive treatment. Detection is via cell culture, primary monkey kidney. Primary monkey kidney cells are used. Yung cell line, PMK cell line. Then you have your enzyme immunoassay and your fluorescent antibody stain. Treatment is supportive. Antivirals such as your amantadine and rimantadine are used. Prevention is influenza vaccine or antiviral prophylaxis. So once it starts, before the rainy season comes, class, I do advise you to have influenza vaccine as much as possible. It reduces the chance of you acquiring this type of viral respiratory disease. Then we have your influenza B. Now, influenza B class is like a mild influenza. So, influenza A is the one that's more dangerous or deadly, while B is a form of a mild influenza. So, detection is via cell culture, primary monkey kidney, enzyme immunoassay, F18. Then treatment is supportive, but you could also use sanamivir and oseltamivir. Prevention is via vaccine and antiviral prophylaxis. Then we have your epidemiology. Antigenic drift only would result in local outbreaks every 1 to 3 years. So in the next slide, I will explain, class, what is this antigenic drift. Yeah. Okay. So for you to understand antigenic drift versus antigenic shift, so let me explain this. Your influenza virus class is capable of becoming or performing this type of change. Now, influenza virus are unique because of their ability to alter their antigenic composition, thus reinfecting non-immune susceptible hosts. So what do I mean by this, class, is that, remember, in your immunology and serology, your body, specifically your specific type, also known as your humoral immune system, is capable of what we call memory. If you remember, your humoral immune system would usually involve your B cells, and your B cells in the long run would become plasma cells, and these plasma cells would produce different types of antibodies. Now, these antibodies, class, have what we call a memory. When you say memory, they're capable of remembering an antigen's characteristic. Now, the problem with your influenza virus, your Orthomyxoviridae family viruses, is that they're capable of changing their antigenic composition. So once they infect the same person, they could cause a reinfection. How? Remember, they would change their antigenic composition. Let's say that your antibodies identified or remembers this person. Let's try to apply this or let's try to apply it to the virus. On the surface of your virus, let's say there would be square square receptors. You were infected with this type of influenza virus on July with square receptors. You were able to recover. Now, later on, the influenza virus was able to mutate or change. This time, after mutating, instead of having square antigenic receptors, they would now have triangle receptors. Since there is a different antigen composition, your body, your body, or your body's antibody will no longer remember this antigen, causing a reinfection. So the body's antibodies will no longer target that virus because they do not remember it, but and that would lead to a reinfection. So let's discuss the difference between drift and shift. So antigenic drift is caused by a mutation during genome replication, resulting in a minor antigenic change and relatively mild influenza outbreak every 1 to 3 years. So to further explain, class, this is the illustration of your antigenic drift. Now, when a virus would usually infect a person, they have the so-called capable of mutation. Mutation are basically changes, class, in their proteins. So over time, the virus would start to mutate. Your influenza virus would mutate based on the replication method, especially in the translation process. Remember, if you remember in the central dogma, translation would produce proteins. So the mutations would occur in their genomic replication, leading to a mild influenza outbreak every 1 to 3 years. So, example of this is H1N1 or the swine flu virus would birds pass it to pigs, then it would mutate in the latter. While the more cause of concern is your antigenic shift. So, antigenic shift is caused by a reassortment or mixing of the segmented viral genome during co-infection in non-human animals. So, when you say co-infection, two viruses class, two viruses, two viruses would infect the same host. And when they infect the same host class, they would combine their genetic material, leading or producing another type of virus, which is the one that is more deadly and dangerous, producing worldwide outbreaks. Co-infection would occur when human viruses would infect a cell at the same time as an animal influenza virus. That's why class your influenza virus nowadays would have an H9N3, H H3N4. So later I will explain what are those H and N. So that's the difference between antigenic shift and antigenic drift. If it's antigenic drift, there's a single virus that would mutate every time it is transmitted to another host. But if it's antigenic shift, there is a co-infection class. Two viruses would have their genetic material combined in a host, producing a more deadly type of virus. Okay. So the Orthomyxoviridae would include influenzas A, B, and C virus. So the virus would contain a positive sense, a single-stranded RNA, and a helical symmetry and our envelope. Influenza virus would range in size from 80 to 120 nanometer. Now, the characteristic of this virus is that it has the presence of two antigens, the HA and the NA. HA would stand for heme agglutinin antigen, and NA would stand for the neuraminidase antigen. So let's discuss them. HA or the heme agglutinin antigen class allows the virus to attach to the sialic acid glycoprotein on the surface of the RBC and respiratory cells. Sialic acid is often found on the surface of RBCs and is responsible, if you remember, for the negative charge or the zeta potential, allowing attachment. So yung negative charge class would repel positively charged bacteria, fungi, and even viruses. Promotes the fusion of viral envelope to the host cell membrane and can hemagglutinate human, chicken, and guinea pig RBC. Elicits the neutralizing antibody response and is responsible for the entry of virus. While the NA or the neuraminidase antigen permits the virus entry into the host cell. So it allows the virus class to enter inside the host cell and cleave the sialic acid on the viral particle, which helps to release the virus from the host cell, responsible for the exit of the virus. Now, it's very important to understand the HA and NA class because remember the number of mutations the one that scientists use nowadays, is that the number of mutations of these antigens would denote the type of influenza. Kaya nga meron silang H1N1. Now, I think there's H9N9 na class. That's the number of mutations that would occur. So the number there would uh, would explain the number, or would identify the number of mutations of those antigen. So if it's H9N9, meaning the hemagglutinin antigen had a total of 9 mutations. If it's N9, neuraminidase antigen had a total of 9 mutations, something like that. So this is a further explanation class of your antigenic shift. So we have migratory birds. So we have here an influenza virus from birds. Then we have your influenza virus from humans. So both of these influenza viruses class would uh, would would stick to the swine. These two viruses from birds and from humans. And when that happens, they would produce a third type of virus, or a third type of virus in the animal, causing a more deadly virus. But if it's antigenic drift, the influenza virus from the chicken would just mutate on its own on the swine. If it's antigenic drift from human, influenza virus from the humans would just develop from, well, would mutate in the swine. There is no co-infection. So remember that class. If it's antigenic drift, there's no co-infection. But if it's shift, it would require two influenza viruses producing a third, more deadly virus in a host. So lab diagnosis is that specimen for collected for identification of influenza virus would include throat swab and nasal aspirates or nasopharyngeal swab. Influenza virus disease is detected in the laboratory by cell culture, fluorescent antibody staining, or other antigen detection method, serological testing, and even Okay, let's continue. Let's go to the fifth one class. We have this time your Paramyxoviridae. So this is a very interesting group of viruses class, because you'll be able to relate to this one. So let's look at the summary. So the summary of these viruses is that the family is the Paramyxoviruses, Paramyxoviridae. The common name is that they are known as the Paramyxoviruses. Now these viruses class are single-stranded. RNA genome has a helical capsid with envelope, not segmented like your Orthomyxoviridae. So under the Paramyxoviridae class, we have different viruses. We have number one, your measles virus. So transmission of your measles virus is via contact with respiratory secretion and is extremely contagious. Diseases caused would be measles, and there's also your atypical measles. So atypical measles would occur in those with waning vaccine, and we also have subacute sclerosing panencephalitis. Detection is again via cell culture using your PMK and serology. Treatment is supportive. Immunocompromised patients can be treated with immune serum globulin. Prevention class, there's what we call measles vaccine. Then we have another type of virus here, mumps virus. Transmission is person to person contact, presumably respiratory droplets. Disease is known, it's known to cause your mumps. Detection is via cell culture and serology. Treatment is supportive. Prevention is via the mumps vaccine. Then we have your para-influenza virus. So for the para-influenza virus is contact with respiratory secretion. Adults would have diseases seen in adults would be upper respiratory and would rarely be pneumonia. Children would have respiratory diseases including croup, bronchitis, and pneumonia. Detection is via cell culture, shell viral culture, and FA stain. Treatment is supportive. Prevention is avoiding contact with the virus. And there are 4 known serotypes of parainfluenza. Diseases would occur year-round. So the Paramyxoviruses class would include the genera Paramyxovirus, Morbillivirus, and Pneumovirus. The family would possess negative sense, single-stranded RNA, helical symmetry, an envelope, and an average size of 150 to 300 nanometer. The Paramyxoviridae family would include many pathogenic viruses, especially for young children. The genus Paramyxovirus would include Newcastle virus, mumps, and Parainfluenza virus. Paramyxoviruses do not have segmented genome and therefore do not undergo antigenic shift like the orthomyxoviruses. The morbilliviruses would include your rubiola, which would cause the measles. The genus pneumovirus would include the respiratory syncytial virus or the RSV. Now, diseases caused by measles and mumps viruses can be eliminated by vaccination. Meron tayong tinatawag, we have the so-called MMR vaccine, your measles, mumps, and rubella vaccine. At an rubella virus. At an early age class, we are given this vaccine MMR, which is being required for all children. Now, mumps virus class would cause parotitis. And what is this parotitis? This is the enlargement or the swelling of your parotid glands. At the age of, if I recall, 16 to 18 class, you would develop this type of disease wherein you would have a swelling of your parotid glands, usually located behind the ears, and you would have a difficulty of swallowing. Now, there's a theory class that if you get your mumps after age 22, it can lead to infertility or you will not impregnate someone. You're considered infertile class yung lalaki. If you're a male and you had your mumps at the age of 22 and above or 21 and above, you would be considered infertile. That's actually true class. There's a journal stating that it happens because the virus would end up going to the gonads and it could destroy your ability to create sperm cells. Now, mumps virus would possess both HA and NA antigen and a hemolysin. Measles or rubella virus possess hemagglutination antigen and a hemolysin but does not possess neuraminidase antigen. Then your RSV or your respiratory syncytial virus would contain a surface protein called your F protein or your fusion protein. Now, F protein would mediate host cell fusion into syncytial cells, which are the hallmark of RSV. If you remember yung syncytial cells nyo class, they appear as clump or fused cells, kumpul-kumpul in Tagalog. So this is because of the fusion protein or the F protein. They cause the syncytial cells to fuse together or to attach to one another, appearing as clump. Now, RSV disease in newborns with underlying medical conditions, especially premature children with developing lung infections, can be prophylaxed with RSV immune serum that prevents severe RSV bronchiolitis during susceptible early months of life. Except for RSV, the paramyxoviruses would absorb guinea pig red blood cells. So all your Paramyxoviridae viruses class are capable of absorbing guinea pig RBC except for RSV. Now, detection is performed using cell culture with hemoabsorption, FA staining, or enzyme immunoassay. And let's go to the last virus class, yung Coronaviridae. So Coronaviridae, under here we have the common name coronaviruses. The target here is your coronavirus class. This is a single-stranded RNA virus with a helical capsid and has an envelope. Transmission is via direct contact or aerosol. Disease that it would cause is the common cold. So the one that causes your common cold class is your coronavirus. And in some cases, it could also cause gastroenteritis in children. Detection is via electron microscope. Treatment is supportive and avoiding contact with the virus. So many coronaviruses of humans and other animals would exist. The prefix corona is used because of the crown-like surface projections that are seen when the virus examined under an electron microscope. So in the surface of your coronavirus class, you would find this crown-like projections. This is the reason why it's known as coronavirus. It has this crown-like projections when you view it under your electron microscope. Now, human respiratory coronaviruses would cause colds and occasionally pneumonia in adults. Coronaviruses are thought to cause diarrhea in infants based on the presence of coronavirus like particles in stool of symptomatic patients. That is why, class, during the COVID pandemic, if you recall, they were able to find traces of coronavirus in sewer or in sewage. The reason for that is that it could survive yung mga projections na yan in the stool of individuals. That's the reason why during the pandemic. Now, the diseases it causes, class. So, one of the diseases it causes is your MERS. Now, MERS is known for your Middle East Respiratory Syndrome. This is a novel disease that was newly discovered in 2012. It was first identified in Saudi Arabia. Now, symptoms would include fever, cough, and shortness of breath. Pneumonia is common but not always present. Gastrointestinal symptoms class would also present, including diarrhea. Some laboratory confirmed cases of MERS-CoV are reported to be asymptomatic, meaning that they have no clinical symptoms class. They have no clinical symptoms, but if you perform an antigen test on them, they are positive for MERS-CoV infection. Now, most of these asymptomatic cases class have been detected following aggressive contact tracing. Approximately 35% of reported patients with MERS-CoV have died. Although most cases of MERS-CoV infections have been attributed to human to human infections, current evidence has classed that there are the presence of dromedary camels being the major reservoir. So MERS-CoV viruses class would usually come, according to this, from camels. So they would be the reservoir. When you say reservoir, that's where they would propagate or they would multiply. However, the exact role of dromedaries in transmission of the virus and the exact route of transmission are unknown. Now, the deadly one, the recent one, is your COVID-19 or your SARS-CoV-2. This is an infectious disease caused by a newly discovered coronavirus. People infected with COVID will experience mild to moderate respiratory illness and would recover without requiring special treatment. Older people and those with comorbidities class, such as your CVD, cardiovascular disease, diabetes, chronic respiratory disease, and cancer, are more likely to develop serious or severe symptoms. Now, this is primarily spread through droplets of the saliva or discharge from the nose. That's why it's very important, class, you would practice proper respiratory etiquette by coughing into an flex elbow. The new strain, so ito yung strain niya before class. The B.1.1.7 is characterized by 17 mutations that would cause amino acid changes, 8 of which would occur in the gene for the spike protein. Nowadays, class, there is a updated list of mutations. Ang daming mutations na ng COVID-19 natin. Now, there are other mutations in other regions of the CoV2 SARS, the COVID genome that would provide a genomic signature for this lineage. A highly transmissible strain lineage designated as B.1.351, also known as your variant 501Y.V2, was recently identified and would have multiple mutations in the S gene. So these are the references. Thank you.